Visualization course for fall 2018. My name is Elham Jafari and my project is predicting breast cancer. Breast cancer represents one of the diseases that make a high number of deaths every year. It is the most common type of all cancers and the main cause of women's deaths worldwide. Screening is testing to find cancer or other disease early in people who have no symptoms. Screening can help find cancers when they are small and have not spread, when they have a better chance of being cured. The early diagnosis of breast cancer victims can improve the prognosis and chance of survival significantly, as it can promote timely clinical treatment to patients. Further accurate classification of benign tumor can prevent patients from undergoing unnecessary treatments. Thus, the correct diagnosis of breast cancers and classification is really important for us. We're using fine needle aspirations, which is a type of biopsy procedure where a small sample of tissue is extracted from a patient in order to make a diagnosis. And then we're using macroscopic images. Biopsy images were digitalized and features were extracted, including radius, symmetry, smoothness, and etc., with target being benign or malignant. We have ID for each patient, and also all the other columns are float numbers except diagnosis, which we will map it to 0 and 1. We're exploring various aspects of the breast cancer dataset to get more familiar with nature. Another objective is finding correlations between different features and diagnosis label. We're also visualizing different features using Python libraries, and also we're using PCA to describe the data in lower dimensions. In the picture we're showing a magnified image of a malignant breast fine needle aspirate, which is visible cell nuclei and are outlined by a curve-fitting program. According to the related work in 2010, Osha Goodwell proposed a wrapper approach with genetic algorithm for generations of subsets of attributes with different classifications such as C4.5 or Bayes networks and etc. Also, we have Abul Ela Hassanian in 2003 who had experimented on breast cancer data using feature selection. In 2005 we had Kemal who proposed a new classification, and also we had Daisy in 2007 who had lots of experiments. We see that cancer cells usually display large variably shaped nuclei. A slide of cancer cell may shape a large number in process of dividing, and in disorganized arrangement also they are darker because of their nature. Therefore, we should be able to see a correlation between features that capture these irregularities and deformations of a cell nucleus and the diagnosis of tumor mass. After the individual nuclei have been defined for each image taken, the imaging software starts to extract 10 different measurements for each nucleus: radius, texture, perimeter, area, smoothness, compactness, symmetry, concavity, concave points, fractal dimensions are these features. Next, because they were multiple cells identified per image, the data above was aggregated using the following like mean measurement, the standard error of measurement and the worst or largest measurement found for every several nuclei per image for every measurement. Therefore, we have 30 features for our data. According to the distribution of the benign and malignant table below, the number of zero values in dataset, which is orange bar in the plot, shows the number of malignant, which is 212. On the other hand, we can see that we have 357 benign, which are shown as the blue bar in the plot. Hence, we have more data on benign cells. Some of the features are significantly correlated according to above with the diagnosis results according to the heat map plot. We can find about 10 features which have correlations more than 0.6 or less than minus 0.6, which is really high. And then we see the most correlated features in the table below, which are concave points worse, perimeter worse, and etc. We see the mean of concave points worse for diagnosis equal to 1 is 2.44, greater than the average value of this feature when diagnosis equal to 0. So this is very important. Also in another plot, we see that the values of concave points worse for diagnosis equal to 1 is significantly greater than the values for diagnosis equal to 0. We're having a swarm plot here of the most correlated features with diagnosis before data standardizations, which doesn't show us really helpful insights. After that, we decide to standardize the data to see the swarm plot of the standardized most correlated features, and this is much more helpful. I can use pair grid plot here to show the correlation between three features that look more correlated. I discover radius worst, perimeter worst, and area worst are correlated as shown below. I also tried exploring with PCA after the normalization of data. In above, you can see the cumulative explained variance for all principal components. According to the plot, our top three features account for nearly most of the explained variance in the dataset. These results suggest a better approach to simplifying our model. We know that Tisane is very good at picking clustering structures, so I'm putting these PCA and Tisane together because PCA is also really helpful because it's simple, deterministic, fast, and highly interpretable, especially with a data manifold which is not complex. The visualizations are more helpful when we normalize the data. Before the normalization, some part of the visualizations, especially swarm plot and also PCA, are vague and inefficient. After normalization, we can claim that top three features account for nearly most of the explained variance in the data, which is not true when we haven't normalized data. In that case, the three component can show us about 73% of the variance in the data. Cancer cells usually display large variably shaped nuclei. They're also darker because of too much DNA within the nucleus, because of the feature of their nature. So we find the correlations, and the most correlated features include concave points worst, perimeter worst, concave points mean, radius mean, perimeter mean, area worst, radius mean, area mean, concavity mean, and concavity worst. We can see that the benign cells have lower values for most of these 10 features. The smoothness underline SE is very controversial. So for the future work, I'm deciding to work on analysis and find more correlations between the features altogether.